Okay, so now we're going to talk about DNA sequencing. Now, DNA sequencing is a process where scientists can determine the specific order of nucleotides of a DNA strand. Today, I'll be explaining the process of DNA sequencing using the Sanger method. To start, we have to have the particular region of DNA we plan to sequence, and then split it up into 4 separate tubes. But to divide the distinct strand perfectly, we need to use PCR cycling to clone the DNA strand sequence so that it will use the same sequence throughout the test. The setup of PCR and the Sanger method are very similar. DNA sequencing uses the same 5 primary ingredients as the PCR cycling does. However, it adds one more ingredient that's very necessary for the process to work, and that is dideoxynucleotides. Now I'll explain this. So, dideoxynucleotides are very similar to regular deoxynucleotides, besides one primary aspect in their structure. Dideoxynucleotides have a free prime hydroxyl group or hydrogen atom, while deoxynucleotides have the hydroxide group. Now this difference of hydrogen and hydroxide atom is very important because it determines whether or not the DNA chain terminates. So here, I'll give you an example. As you can see, this is a normal deoxynucleotide because it has a hydroxide atom. And here, the hydroxide atom has a oxygen atom where the phosphate group of the next nucleotide can attach itself and continue the chain. However, if the next nucleotide has a hydrogen atom, it doesn't have that necessary oxygen atom, so the phosphate will attach itself. Therefore, it's stopping the chain of DNA. Now that you have a better understanding between deoxynucleotides and dideoxynucleotides, we can continue with the DNA sequencing process. As I said before, PCR cycling process and DNA sequencing method are similar with the ingredients that are used for the 2 operations to work. However, we are going to separate the original copies of DNA into 4 different test tubes. So remember, all 4 test tubes have the same 5 ingredients, but add dideoxynucleotides. However, you want to add one specific dideoxynucleotide to only one tube and not all of them. So, if this was a dideoxynucleotide that represented adenine, you'd only add adenine dideoxynucleotides to this tube. Then, if this dideoxynucleotide was cytosine, you'd only add cytosine dideoxynucleotides to this specific tube only. And the same goes for the next 2 test tubes. So the PCR cycle goes through the 3 core stages of denaturing and annealing and extending, so the original template of DNA has millions of copies. So remember, denaturing stage heats up the double-stranded DNA so that it could separate into individual strands. Then, at annealing, primers are added and attach themselves to the complementary strand of either side. Then, the Taq polymerase atoms bind themselves so they can extend the nucleotides and create a complementary strand. This one. The same thing goes for this side as well. When extending the DNA strand, it will produce a new strand like normal by adding complementary nucleotides, as I showed. However, the chain will terminate at different lengths whenever a dideoxynucleotide is added each time. So, if we were to draw this out, say this green line represented the one of the strains of the DNA copy. And if we were using the adenine dideoxynucleotide, let's see, and if a dideoxynucleotide was added here, because that's where one of the adenine molecules would be, there would only be one strand of DNA that long. And say it makes another copy of DNA to complement it, but then it stops only here because that's where the next adenine dideoxynucleotide stops at. And it keeps going down the chain depending on where the next adenine nucleotide is. So we will have different lengths of DNA fragments produced. Once millions of copies of different sized DNA sequences are created, we identify the actual sequence itself through gel electrophoresis, where the DNA fragments move through a gel that will separate each segment from largest to smallest. So let's say this is a field of gel electrophoresis. And since we had 4 different test tubes, we're going to have 4 different lanes for each test tube. Now, at the top where the DNA samples are placed, it has a negative charge. Whereas the bottom portion right here would have a positive charge. So, since the strains of DNA have a negative charge, the positive end of the gel electrophoresis field will attract the DNA strand and pull it downward. However, this will help separate the different DNA strands by length, because the larger the DNA strand, the smaller the DNA strand, the further down it will go, and the faster down it will go. However, if it's larger, the largest DNA strands will stay here and then get smaller as you go down. So, if we were testing the adenine fragments first, we place all the adenine fragments here, and then as they move down, one by one, the larger one will stay closest to the negative side, and then as the smaller ones keep going down, the smallest will hit the very end of the electro field. And the same goes for the thymine nucleotides. As you can see, we'll start here and then go down. And the larger fragments of DNA will stay while the smaller ones keep moving closer and closer to the very bottom. And the same goes for the cytosine and guanine dideoxynucleotides. Okay, so here we have the finished gel electrophoresis test, and each lane has the different fragments of DNA in a specific order. So we can start merging each lane together depending on its order, so we can identify the sequence of the DNA nucleotides. And once we're done putting all the lanes together, it'll create the specific sequence we had at the beginning. So we have correctly identified the DNA nucleotides.